Hey everyone, this is Dr. Jyoti Bala. Today in this session, I will be speaking about a very important topic, which is drug designing and development. So if you are a young student or researcher who want to explore and come to this in silico drug designing or molecular drug designing or any kind of therapeutics, the subject area is very relevant for job purpose also and for research purpose also. So we will be covering an overview and conceptual detail, what skills and what stages it's required. So let's begin the session. So before coming to the technical part, I wanted to emphasize one very important point over here, especially for the student and the young researcher who are planning to come to this sector. Any drug designing and development projects overall requires a multiple interdisciplinary kind of team. It's not like one individual scientist or researcher should know everything, but it's good if you know multiple skills. So people from biology, life sciences, biologists who understand cell biology, microbiology, molecular biology, genetic, immunology, biotechnology will be required to understand the diseases, its causes at molecular level and cellular level, so that we can get the specific targets and when we can develop a diagnostic, therapeutics and vaccines for that. So at molecular and cellular level, you have to understand the diseases first, and then either you are designing a drug based on nanotechnology, molecular chemistry based drugs and so on, you will require these studies at the chemistry level, at the toxicology level, at the pharma level and so on. And in these days we can do in silico drug designing, so computational biologists, biotechnologists, bioinformaticians will be required. So this is the reason I was saying we require interdisciplinary kind of people. So either if you are a student, you enhance your skills in multi-dimensional subjects, or maybe if you have certain interest, you can enhance the skills and then come to these kind of sectors and you can start these kind of projects. It is not always required that you know 4 or 5 skills or 4 or 5 subject area, but it is always good to come to this sector if you have additional and multiple interdisciplinary kind of skills. Like in my own case, I started from botany, but then I did my PhD from biotechnology related subjects and then to nanotechnology, drug designing, and all these things I have learned during my PhDs and postdoc experience. So here also in drug designing thing, people for understanding the diseases, pathogen, drug and target interaction studies, and then finally the delivery of the drug in a safe way will require these kind of different people from biology, chemistry, nanotechnology, data sciences and all those things. Okay. So let's discuss a little bit about the drug designing and drug discovery basics. We all know that we require drugs to prevent and treat multiple diseases. We all are seeing this COVID period and how challenging the things are for everyone globally actually. So if we talk a little bit about the traditional time, during that time mostly people were dependent on random and traditional approaches like natural therapeutics and all these things. And many a time via serendipity also some drugs came to the market also. But now we are in a modern time and we have seen technology advancement both in dry lab and in wet lab, which is actually helping us a lot. In the past, drug designing and development used to take 10 to 15 years with interdisciplinary team and a lot of money. But nowadays we are in a better time, and in COVID period also you have seen how many diagnostic, how many vaccines came in a small duration of time also. That has happened because technology has advanced in biological sectors, in dry lab, in bioinformatics and in multiple sector actually. So via using modern high throughput screening, in silico approach, how we can efficiently design and develop drug, we will be discussing all these things. So we will get the ideas what approach and what technology people are using these days to smartly understand the diseases, to finding out the right targets, and thereby designing a screening and assay methods and developing an overall drug effectively. These are some of the top global pharmaceutical company. You can see it's a already billion-dollar industry. Companies like Pfizer, Novartis, Takeda, these are in the top 15 list always. So if you are planning to apply globally in these sector, you can approach these companies. Similarly, if you are planning to apply in Indian pharmaceutical company, Cipla, Dr. Reddy's, Sun Pharmaceutical, these are some of the best pharmaceutical companies where you can apply. So let's briefly discuss about what drug designing and development is. As I have already mentioned that it requires interdisciplinary experience and knowledge in team. And then either you are using random approach, de novo approach, high throughput screening or computational based approach for designing and discovery. One thing will be to understand the disease, and based on that we have to find out the molecular targets which are responsible for that disease, and then we have to find out some lead candidate which can modulate and inhibit the target properties, and then finally lead optimizations to safely deliver these drug candidate after checking the pharmacokinetics, dynamics into the human system. So when you are choosing any CAD or drug designing and drug development related project, we have to understand the diseases first. As I already mentioned, disease can happen because of multiple reasons. Sometimes it happens because of microorganisms, the pathogens like parasite, protozoa, fungi, bacteria, viruses, and in that pathogen itself there are different kind of pathogens. Suppose if you're handling virus kind of antiviral project, so your DNA viruses are different than your RNA viruses. In RNA viruses also your HIV is different than your SARS-CoV-2. So at molecular level, how these pathogens behave, what's their physiology, what's their way of infection, how it is going to interact with host, all these understanding is very important, and based on that we select the disease model, we select the strategy of developing these drugs related to that particular diseases. So whether we are targeting any biological disease or genetic based on mutation or genetic rare diseases, things will be different. Although we are designing drug, but because the causing agents are different, the mechanisms are different, our strategy of developing a drug will also vary. Similarly, if you are handling a metabolic disorder like diabetes, there you have to handle differently that disease project. Okay, and sometimes we have to develop a drug related to autoimmunity or autoimmune diseases, and then sometime we have to make disease related to pain and all these things. So what is our purpose, for what purpose we are generating drug is important. And then there are these infections. If you are handling a pathogen related drug, then either the infection is acute infection, chronic infection, latent or reservoir. Things are different in different microorganisms. So those challenging point we have to address via our strategies when we are developing any drugs. So if you will see the overall drug designing and development process, it's a lengthy process. It requires interdisciplinary team, and depending on our skills, our interest, we can contribute at any stages. Like if you are a molecular biologist, initial understanding about the disease, you can participate in developing the bioassays. If you are a clinical research scientist, you can work in this sector. So whatever stages and interest you have, you can plan out how you can enter the sector. If we talk about the overall durations and the time span what is required while developing these things, in past it used to take 10-15 years, But as the technology has already advanced, so nowadays this duration has been reduced. Now within 5, 6, 7 years, we can get drugs, but it's a complex procedure. We all know that part. Like in past, people in pharma and in laboratories also, they used to screen 20,000, 50,000 compound, and few candidate only get approval after preclinical, clinical studies, and in between, in during the preclinical studies or in vitro, in vivo studies or pharmacokinetics, pharmacodynamic, all those studies we have to perform to check the activity, to check the mode of action, to check the pharmacokinetic dynamics and all these things. So it's a lengthy process. Usually in past, a lot of candidate we have to screen, and only few candidate gets approval. But now we have to reduce these numbers of candidate. So if you can see this tunnel, we have to reduce these things, so that we have to screen a right candidate for a particular disorder. And with in silico approach and with other technologies, we should like send right candidate to the next level, so the chances of getting the approval will increase if we do smartly and logically each step. And here your in silico approach, your high throughput biotechnology related assays development and technologies are actually helping us to develop a drug, and also in vaccine development sector also. So in summary, you have to actually find out, if you are new, you have to understand the diseases, its challenging issues, what target it is causing that particular disorders. Either we have to inhibit or activate that molecule based on whether it is harmful or beneficial for us. Like in case of insulin, we have to produce the recombinant insulin so that it can protect the diabetic patient. But suppose in other cases, like if you are treating a cancer patient, like BCL2, EGFR, these things causes cancer, oncogenes. So here we have to inhibit and modulate the activity of those proteins. And similarly, not only inhibition and modulations we have to perform, we have to prove the mode of action of our drug. So in that case, we have to prove the activity of our drug. For that, we have to design bioassays also, and whatever leads and drugs we are testing in this disease models, we have to optimize them for the better efficiency, better delivery, specific targeted delivery, and so on. So overall, these things we have to take in considerations, and the drug and the target interaction is based on structure. So a lot of in silico structure characterization, and in wet lab real-time structure experiments also we have to perform to prove that these targets are binding to this ligand, and specifically they are modulating and having some therapeutic activities. So all these experimentation we have to perform to bring these candidates in the clinical testing also, and to the final to the market also. Here, a few things also I wanted to highlight, which is very important actually. We are in a time of big data biology. Data is playing a huge role in every sector, and similarly in drug designing and development, and in biological sector also. Because we are in a time when we can generate a lot of genomics, proteomics, metabolome, microbiome, and all sort of omic data. So in a biological sector nowadays, we can understand any disease in a much better scenario these days. And similarly, we can understand the host, genetic makeup, and all these things. So in a system way, in an integrated way, these days biologists and drug designing people also require to understand these amount of data, the omic data, so that things can go in an efficient way. And this is the reason we are seeing the time of personalized medicine and precision diagnostic. So smartly utilizing the biological data to understand the diseases and to pick the right ligands and effectively design and develop these strategies. So using the biological big data will be very important in drug designing sector also. Now the other term, like as I said, the multi-omic. So to understand your host, to understand a disease condition or the overall physiology, we have to understand the DNA whole system at the genomic level, at transcriptomic level, not only the molecular things but how outer things affect to us. So epigenome, what microbiota we have, so overall microbiome. So in a omic level we have to understand any particular diseases and in an integrated way find out why that disease is happening, and based on that rightly pick your targets and then design your drug designing and development strategies and projects. So in pharma, in biology, in biotechnology, these days people are required to understand and analyze a big data related to biology, to understand the diseases, to understand the host, to understand the host pathogen, how our immunology, how our microbiota affect us. All these concern areas in an integrated approach way we have to understand. And if you are from biology, if you are from biotech, if you are from pharma, you have to make yourself comfortable with bioinformatics and certain Python, Linux are related things. And if you want to work in such kind of project in a customized, in a automated way, then in near future, not in near future, actually in drug designing people are using AI and all these things. So if you want to come in the advanced stages, then starting from your understanding about the bioinformatics, computational biology, Linux, R, Python is going to support you a lot in this kind of work in an automated, in a customized way. So these days in drug designing, people are using AI, machine learning and all those things. So while doing these kind of projects and work, a very important part after your understanding about the diseases at molecular level, we have to rightly pick a target which is causing a diseases. In past, if you will see the literature, mostly people has targeted enzymes, protein, hormones and all these things. But because of omic data, nowadays we can target genomic, transcriptomic, metabolome and all these integrated approach. So on totality, omic data we have to consider. So based on that, we have to pick the right target which is causing the disease, and thereby we have to design the drug strategies based on that. So when we are doing this target identification, next step is target validation, which is also very important. Whether in wet lab and with dry lab experiments, we have to find out a right target. So validation in terms of its overexpression, knockdown. So to 100% sure that okay, this protein, this metabolite is responsible for that particular disease condition. So here your molecular biologist, biotechnologist, biochemist, cell biologist does all those tasks in wet lab and your bioinformatician in dry lab. So other additional experimentation will be required so that we are 100% or maybe we are sure that okay, this is the right target which is causing the diseases, and now we want to inhibit and modulate these things via our ligands and our drug candidate. So target identification and target validation both are important. So if you want to use computer aided drug designing or any in silico approach for your drug designing and development, a very renowned approach is computer aided drug designing, which is also known as CADD. Many of my students and researchers recently who has done their dissertation and training under my guidance, few has got US scholarships also, and one has got Max fellowship from Japan also. So if you learn this kind of approach, you can utilize these approach for multiple disease and different kind of project. In CAD, it can't be taught in 10 minutes or 15 minutes. It's an overall database and technology based approach. So we have to provide some time over there. In upcoming videos, I will be providing you in detail how CAD can be performed. But here you have two approaches usually, like structure based and ligand based. here mostly we have to work from structure level of your targets, the proteins, and we have to then do the ligand docking and all these things. Whereas in ligand based computer aided drug designing, from ligand we go to target, from ligand structure we predict the target activity, and we perform SARS and quasar. So here both little bit the approach is different, but by using computational tools, databases, we come and we find out certain target and ligands, and we check their interactions in different approaches in different way, and basically we design a drug, and we also predict physiochemical properties, we predict their interactions, their affinities, so that a right candidate we can send to the preclinical testing level and for structure characterization and in vitro in vivo level. Not only your in silico approach, but as I mentioned, in drug designing and development, a very important part will be to validate and to tell the mode of action of your drug also. So a lot of bioassays also we have to perform while doing such kind of project. So either in lab condition, in vitro, or sometime by using animal model, in vivo experiments you have to perform, sometime using high throughput screening or real structure characterization, either by surface plasmon or via radioactivity. You have to tell what is happening because of your drugs and how your drug is inhibiting and having that therapeutic activity. So different kind of experiments and technology we have to utilize, and we have to develop our bioassays so that we can show what is happening and why that therapeutic activity is happening. So a very important part will be of such project will be your structure studies and structure characterization. Many a time you have to utilize computational databases and tools to get the structural data from databases, and then you have to either sometime you have to model the structures of your proteins or your DNA, RNA, sometime metabolite, chemical, and you have to do certain tasks related to molecular docking and all, and sometimes real characterization in terms of binding, affinity, structure and conformation changes. So real-time structure experiments also and bioinformatics based experimentation also you have to perform to explain and to demonstrate that your target, which is responsible for the disease condition, is inhibited and modulated via your ligands and hit and the drug candidate. So in drug designing and development and discovering part, a very important approach which at molecular level also and at chemical level also people use is combinatorial chemistry. So we have random approach also and combinatorial chemistry based approach also. In fact, the aptamer strategies and the aptamer technology which I use at molecular level for DNA RNA related therapeutic work, there also we make random combinatorial libraries. In drug designing and pharma, people either uses random library, so different chemical compounds, ligands, they test for inhibitions and for modulations of the target via random approach, and sometimes the backbone structure will be same, but different combination of the same chemical compound will be generated. So either they try random libraries, sometimes they make their own combinatorial libraries. So different library generation, or sometimes we have to purchase the libraries, and sometimes we have inbuilt libraries. So depending on what stages, what places we are, and for what purpose we are using these libraries, things will vary depending on your strategies and disease condition. So we have to pick those random libraries, we have to try them for the target inhibition and modulation, and then you have to, if you found out a best candidate from there, then you have to optimize those things for final delivery purpose and all these things. So overall combinatorial library based strategies people still uses. And while doing especially the computational approach, and CADD in silico approach, a very important part will be your molecular docking. So once your target is finalized, which is causing the disease, you know the ligands, you have to check their interaction on the computational platform. So whether you are a beginner or if you have prior understanding, you can use either server based platforms or you can advanced tools also you can utilize, and then you can check these interactions and you can get the ideas about the interactions of your target and ligands, what interaction is happening, what kind of bond is formed, what is the affinity, and all these calculations we can perform via molecular docking. And if you are good in these things and if you know those molecular docking and computer aided drug designing, then there are platforms like Pyrex and other platforms where you can perform virtual screening. So one target with multiple ligands, also one ligand with multiple target in a molecular docking we can perform, and we can get the ideas that what are suitable for binding purpose and for inhibition purpose. So the right candidate we can pick from these virtual screening and send them to the preclinical testing or in vitro testing and the for structure and the for wet lab based experimentations. So these are few of the in silico approach based characterization. See here in COVID-19 proteases has been inhibited via natural therapeutics. So different candidate and phytochemical has been isolated and they have been docked for with the COVID protease enzymes, and they have checked the molecular docking and thereby they have found few candidates which are inhibiting these enzymes which are crucial for the SARS-CoV-2. In different studies they have also utilized molecular docking. So in the COVID-19 we know this spike protein is very important, and then RNA dependent RNA polymerase is also very important because this is a RNA virus. So here they have targeted this RNA dependent RNA polymerase via different drugs which are actually for RNA viruses, and these drugs they have targeted and inhibited, tried via molecular docking whether they are having some molecular interactions, and then they have found which is the good candidate, possible good candidate for the next round. And once your molecular docking virtual screening has been characterized and you have enough data, so from there a right candidate you can send it for the experimental studies where you can check the conformation changes, your real-time binding, your binding pocket with NMR, crystallography, or spectro surface plasmon resonance will give you the real binding experiments. If you want to check the binding pockets and the conformation change, you can perform circular dichorism, ITC experiment. So these kind of experiment, depending on our needs, we can perform and we can validate the binding things related to our target and our drug candidate. Then comes a very important part in any drug designing and development, which is pharmacokinetics, and we have to calculate the pharmacodynamics also. Here we have to actually validate and we have to estimate the ADME, that is association, dissociation, metabolism and excretion related to your drug, and here you have to estimate and you have to find out the mode of action and toxicity related things. So all these experiments in wet lab and dry lab, these days we can perform these analysis and estimations. So pharmacokinetics and dynamic will be important part of your such kind of projects. One more thing is very important while performing computational drug designing and drug discovery and development. Here from SARS or Quasar, from the ligand structures we get the idea about the activity, the possible activity. So we have certain tools and algorithms and databases for that which can do those tasks. So the quantitations and the prediction we can do via Quasar. So you can utilize that also if you are coming from ligand to target and you want to get the ideas what activity that drug candidate could have. So once you have done your projects related to drug designing and discovery, you have to send your right candidates for clinical studies, and depending on which country we are coming in, we have to perform some clinical 1, clinical 2, phase 2, phase 3 trial, and somewhere for phase 4 also. So once everything has been done in lab also and in the clinical research also, when you have checked the testing and the safety of these drugs into the human populations also finally, so you send these things for getting the approval, and depending on which country we are coming from, we have different agencies, and from there we have to send all these things, and you get the approval based on the data and all the experimentation and clinical research data and your control experiment and overall project data. So as I mentioned, in India we have CDSCO, in USA we have FDA. So they will check overall data and the qualities, and based on that the final approval we get. I hope the session was relevant and informative. If you have any queries, you can drop me a message in the comment section, and if you have liked the information, I request you all to kindly like and subscribe the channel also, and to share these information among your scientific endeavor. Thank you so much.